I would like to introduce Hazal Kandemir with her overview of molecular phylogeny of Onygenales. So please take your place and enjoy being with us.
Thank you and welcome back, everyone. I'm very happy to be here with you and to share my presentation, which is entitled Molecular Phylogeny of the order Onygenales. This project was started at Westerdeck Fungal Biodiversity Institute, and it continued with the collaboration between Çukurova University from Turkey and also Radboud UMC from the Netherlands. So as we all know, member of Onygenales can be found on many different kind of sources, like dung, feather, soil, skin, nail, hair. So they can also cause superficial or systemic infections. And first identification of these members of the order was based on morphology of ascomata, ascus, ascospore, pigmentation, presence, and also type of anamorphs. And since then, many different families were described in the order, and most of them were abandoned. Now we are talking about only 8 different families in the order. So what was our aim with this project? It was to provide a framework for the order and to redefine the genera and the families in the order. And also we would like to see if there's any correlation between phylogeny and the other data of the species. We also want to contribute to stabilize nomenclature by indication of mostly missing type or reference materials. So the main question, which came from Sibron, was can we define the order, the families in the order, based on their ecology? If we think their ecologies as entities of evolution, is it possible to show them separately instead of branches in the phylogenetic tree? So we got to work, we searched different databases, and we make a list of species which were described in the order, and we also check for the type materials and strains with the ecological data. And also, with help of Carolina and Benjamin, we checked if there's any available sequences, mostly LSU and ITS first. And if there is no data, we checked CBS collection. And when it's possible, we obtain the strains and perform the PCR and sequencing. And if there's already available sequence, then we check their quality and include them, the phylogeny and ancestral reconstruction analysis. So in the end, totally 521 strain. It's not going, yeah. Totally 521 strains included. And we decided this number basically type species of each, type strain of each species, and when it's available, we took the species, same species from different hosts. And in the end, we had 403 LSU sequences, 481 ITS sequences. And also we saw when we used different number of strains and different algorithms to construct phylogenetic trees, they showed different topologies. So we had to decide which tree would be the main tree to compare our data. So we calculated supported, unsupported clade ratio, and also we made outgroup analysis. And as a result, as you can see here, the ratio for LSU sequences, LSU loci was better than the others, combined or ITS. So we decided to continue with the LSU loci. And the other thing was outgroup, which species would be outgroup. So we tried different species from different orders, also the species in the same order which showed longer branches in the phylogenetic tree. And based on this result, Biciclomys nivea was the best one to use when we checked the clade bootstraps and also supported unsupported clade ratio. So we decided outgroup, we decided which loci we are going to use, and in the end, when we combined these just type strains of LSU loci, we detected 9 supported clades. And we combine it with the ecology data. These colors represents soil keratinophilic, soil oligotrophic, skin, nails, dung, agricultural, hair, feathers, insect pollen, deep infection, plant-based, holophilic, or other unknown. So we decided these categories. And based on that, we saw the strains are mostly from oligotrophic characteristics. And they were isolated from soil, followed by skin and nail. And for this, when we look at this graphic, it was kind of obvious. We can say some clades has kind of certain tendency for ecology, but not all of them, which are here. It's a bit difficult to estimate for clade 2, 3, and 4, basically, main ecology. So we also continue for family by family. And the first clade members were introduced in Arthrodermataceae family. This work already done by Sibrin and other colleagues, and the topology of the tree was not different. But when we combine the other species described in this family, we saw there are some misidentifications, as in here, Gymnoascaceae postea, and also 3 Arthroderma species were recorded as Onygenaceae corvina. And also, there was a problem with the Pectinotrichum because the type species of the genus, Pectinotrichum liense, clustered in ITS and with the other member of the genus, Pectinotrichum chinense. But for LSU, they were different. They were in different places. But unfortunately, this is one of the genera that we don't have much strains and isolates. And for the ecology, strains isolates were mainly from skin and nail, followed by the soil. And the clades between 2 and 4 were just left as incertae cedis. When we are deciding these clades, we just look for the type species of the genera. And then we check clades which this type species in. So we said this clade is Onygenaceae, this is Arthrodermataceae. And this part were not included any of this group. But when we check the other previous studies, we will see many of these species are described as the clade of Onygenaceae, or again incertae cedis. Even this part was described as Oxortraceae before. But when we check, it's because of Oxotron, but then Oxortronopsis was described, and it was showing very similar morphology, but molecularly they are far. And ecology of this group showed they are represented by almost all kind of group that we classify. It was, here you can see only insect or pollen related isolate is not there. And we have totally 4 plant-based isolates, and 3 of them is in this group. And Onygenaceae, based on the type species of Onygenaceae equina, showed mainly from soil isolates. And here you can see there was one specific group here, isolated from skin and nail, skin mostly, and from reptiles, and which was also introduced as Nannizziopsidaceae, as a new family. But based on our criteria to decide clades, we kept it in Onygenaceae instead of separate family. And the clade 6 was represented member of Gymnoascaceae, which is one of the largest group of the order. They are characterized by the yellowish to brownish Gymnoascaceae, as you can see here, with or without appendages. And the differentiation in the family is based on Ascospheraceae morphology. And based on morphology, of course, there are many genera were described. But in 2002, Arachnomycetaceae, Gymnoascaceae, Gymnoascaceae, and Nannizziopsidaceae were classified as Gymnoascaceae based on ITS data and with 23 or 29 species. But when we include all the species described in the family and even in the order, we saw these 4 different clades. And here, ecology again was mostly based on soil, but oligotrophic soil. And there were this group showing halophilic characteristic. And these strains were mostly identified, isolated from cheese samples. And the clade 7 was Ascospheraceae. Yeah. And this clade, this family, is kind of unique because its entire life cycle is in the nest of bees. It's related with bees and pollen. And it has a unique cyst-like fruiting bodies, which is called spore cyst. And this spore cyst includes spore balls, and the spores in the spore balls are important for their classification. And as you can see here, this structure is kind of easier to destruct and disperse. So maybe these reduced fruiting bodies could providing a better adaptation to this habitat. And in this group, 2 species, Apis and Aggregata, they are important for commercially because they're infecting bees mostly. Otherwise, the group members can be found as saprophytic or pathogenic. They are mostly related with solitary bees. And the 8th clade was Spiromastigaceae. It also has orange ascomata, but it has curved appendages, as you can see here. The importance of this family is the members of this family cellulolytic, or they are very weak keratinolytic. So by now, only 3 genera were described here: Pseudospiromastics, Sigleria, and Spiromastigoides, which is known previously as Spiromastics. And the difference between Pseudospiromastics and Spiromastigoides is based on morphology. But between Sigleria and Spiromastigoides, it's based on neutralization. So when we compare the ITS and LSU loci with this family, Pseudospiromastics based on ITS placed among the Spiromastigoides species, but based on LSU, it was separate branch. And there was also Polytolipa hystrix. ITS loci results showed it's in this family, but yeah, for LSU, it was different. It was in different place. It was close to clade 7, Ascospheraceae. So again, we need more isolates to decide phylogenetic position of this genus. And ecology of this species was mostly halophilic. Yeah, here. The isolates were mostly from house dust, and only these 2 species recorded from infections. And these 2 species has a bit difference from the others. They all have hemolytic activities, but these 2 species also has urease activity. And the last clade was Ajellomycetaceae, and this is known with the systemic infection agents in the order. And yeah, as you can see, they were mostly from deep infections. But there was one, as you can see here, there is one strange branch which represented by Arthrodermataceae and Eramascaceae albus. So they were based on ITS and LSU. They are in the family. But this part, Eramascus, was described as a separate family, as Eramascaceae before. It has an intense tendency to grow in a high sugar content. And it also has a different kind of ascomata structure, naked. And Arthrographis has an arthroconidial structure, so they are different than the rest of the family. And there is also Helicocarpus, and which has coiled appendages. And we also saw here Onygenales was with outside of the family, which was described before in the family. So maybe especially Onygenales terrestris can be kind of ancestor of this family because it has no dimorphism. It can grow with maximum higher temperature, and it was isolated from only soil by now. So we don't know about yet Eramascaceae. Should we include it there or it should be separated? It seems it should be separated, but we need more data about that. And in the end, when we summarize the results, we can see keratinase activity is getting decreased, but cellulose activity is getting increased through the bottom of the tree. And we can see different kind of ascomata, Gymnoascaceae or Clastotaceae in the order. So we can say different topology of phylogenetic trees suggests that the evolution in the order occurred in a very short time. And as I show, according to LSU phylogeny, the Ajellomycetaceae is at the bottom. So it suggests it might be ancestor of the order, which is not very, believable. So we need to work on it. And we have some limitations because not all of the genera described in this order were represented by type species. There are some missing data. And as a result of using different families, and the sequence length was shortened during the alignment. And also, classification and nomenclature for the genera which were previously described polyphyletic, like Onygenaceae, needs to be investigated with more data. So we can say, for now, order contains 6 families and 3 clades, and keratinase activity is decreasing while the cellulitic activity is increasing. And we can say some families can be classified based on their ecology. And when we combine the ecology, ascomata, morphology, and phylogeny data, we can define them. So thank you for your attention.
Thank you, Hazal, for the great presentation. We have some time we have one question. And I didn't want to interrupt you earlier. My mom always told me not to interrupt ladies when they talk, so I hope that we are in time. So please, would it be a good idea as a next step to perform mating experiments to find recombinational, and we do not see more. Recombinational, can we see the end of the sentence of someone who was earlier than me? To read it again, please. To read it again, please. Say, ne. Hazal, did you see your question? All right, so recombinational signals with genera to obtain species boundaries. Is a question?
We can try that. Actually, with this data, there is still a lot to do. We can try mating experiments, or we can try to select type species from each genera, because some of them already has full genome sequences, so maybe it can give us more information. But for some species, I'm not sure if the mating experiments would be an answer. But of course, it can be.
Does anybody have a question? From audience, maybe? From audience? No? So thank you very much once again.